Pharmacology is the study of the interaction between a living biological system and a xenobiotic. A xenobiotic is any chemical agent that is foreign to the body, such as a pharmaceutical drug or a pesticide. In 1913, physician Paul Ehrlich established the rule that governs these interactions. Substances do not act unless bound. This observation forms the basis of modern receptor theory. Predicting a therapeutic outcome requires dividing this complex interaction into two distinct measurable vectors. The first vector is pharmacodynamics, or PD. This measures the biochemical and physiological effects the drug exerts upon the body. The second vector is pharmacokinetics, or PK. This measures the actions the body takes upon the drug, how it absorbs, distributes, and eliminates it. Mastering clinical pharmacology requires mapping both the molecular binding of a drug and its systemic journey through the body's geography. This dual vector framework provides the clinical basis for predicting how a specific dosage will perform within a patient's unique physiology. Pharmacodynamics begins at the molecular scale, where the drug acts as a physical signal. Receptors are specialized proteins acting as precise signal detectors. These detectors are stationed on or within a cell, waiting for these chemical xenobiotics. Affinity measures the strength of the drug-receptor chemical bond. Intrinsic activity is the bound drug's ability to initiate a cellular response. Full agonists maximize both. Antagonists have high affinity but zero intrinsic activity, blocking the receptor. Partial agonists only produce a submaximal effect. The precise classification of a drug's affinity and intrinsic activity establishes its foundational mechanism of action. When a full agonist binds to a receptor, the physical event immediately alters the interior of the cell membrane. This initiates signal transduction. Signal transduction is the process where the drug-receptor complex triggers a cascade of internal second messenger effector molecules. This biochemical cascade translates a microscopic chemical bond into a macroscopic physiological effect. Consider the use of epinephrine in local anesthetic agents. Epinephrine binds to its receptor on vascular smooth muscle. The resulting transduction cascade causes the muscle cell to physically constrict, producing localized vasoconstriction. A drug's overall clinical efficacy is limited by optimal receptor site occupancy and the structural integrity of these internal signaling cascades. High pharmacodynamic efficacy is ineffective if the chemical cannot reach the target tissue in the correct concentration. The LADME pipeline is the blueprint of a drug's life cycle. Liberation separates the active ingredient from its formulation. Absorption crosses membranes into systemic circulation, though intravenous injection bypasses this. Distribution disseminates the substance throughout the body before excretion permanently removes it. Tracking the LADME timeline allows clinicians to predict the geographical location of a drug at any given moment. Between distribution and excretion lies metabolism, or biotransformation. This occurs primarily within the hepatic endoplasmic reticulum of the liver. The hepatic cytochrome P450 enzyme system, CYP450, drives this process. It is a family of enzymes responsible for phase 1 oxidation, reduction, and hydrolysis reactions. The chemical goal of metabolism is converting highly lipophilic or fat-soluble drugs into hydrophilic, water-soluble metabolites, so they can be effectively excreted. This graph plots plasma drug concentration against time, mapping systemic elimination. The steep alpha phase represents rapid distribution into tissues. The gradual beta phase slope is driven by metabolism and excretion. Its speed is measured by half-life, the time to remove 50% of the drug. Mapping this biphasic curve is necessary for sustained therapeutic treatment. Supplying a dosage that exceeds the elimination rate leads to enzymatic saturation and systemic toxicity. Safe clinical practice requires calculating the mathematical intersection of the pharmacodynamic and pharmacokinetic vectors. Pharmacodynamics establishes the minimum effective concentration, the biological floor required to trigger a cellular response. It also identifies the threshold for toxicodynamics, the biological ceiling where adverse and harmful effects occur. This graph brings the vectors together to reveal the therapeutic window. This is the narrow zone lying between optimal efficacy and toxicity. Pharmacokinetic principles dictate the dosage and frequency required to maintain the plasma concentration curve within this shaded band. Pharmacology functions as the engineering of drug concentrations at the site of action, ensuring a predictable biochemical response within safe metabolic limits.